So, um, how are you guys doing? This is our lecture for transcription and translation. Those are the two official terms for the steps of protein synthesis. And we haven't exactly heard those terms before. You guys have been looking at those processes, and I'm going to try to help you understand today why those processes are called those names, transcription and translation, and what those processes are. We've already, started looking at that, but today is about sort of breaking it down, understanding what does what, when, and why. All right, so let's start. This lecture is not short. It's fairly long, and you should go into it knowing that I hope that you will keep in mind it was originally designed as a pair deck. So I do expect you to try to answer the questions that are asked or make note of any questions that confuse you so you can talk to me about them. This is the kind of information exchange that is intended to not be passive. Okay, you shouldn't just be sitting there zoning out because that's pointless. Okay, so let's get going. Look at that beautiful orange. Did I already say that? Okay. So here's a review. It's a pair deck slide. You should know this by now. What bases pair with what just in DNA, right? A with T, G with C. And then we haven't talked super exclusively about a whole lot about like cell classification. Our cells are called eukaryotic. The other type of cells are called prokaryotes, although that's not exactly correct. There are other things out there, but this becomes important because in our cells, and of course we're focusing on humans, right? We're the only important things in the world. Haha, just kidding. But when we look at protein synthesis, where the DNA resides, which is in the nucleus, is very important. Okay, so that's why I've asked you this question. More on that later. Okay, so here's an overview. I like to begin with, okay, what's the broad picture of what's going to happen? This is a very busy picture. This is nothing I would ever expect you to absorb right from the very beginning. But it does show from step 1, 2, 3, 4, 5, what happens in the transfer of information from DNA into a messenger of RNA, and that messenger delivering that message to a ribosome, which is a protein factory, and other types of RNA helping to build a protein. So that's what we'll be talking about. All right. So let's look at words. Words. I love words. You can actually really help yourself by attempting to understand why we use the words that we use. We don't just arbitrarily use words. We use them on purpose. Okay, so someday you'll graduate from Punahou, and your transcripts will be sent in fact, before you graduate, you'll be applying to colleges. You'll be sending your transcripts. So your transcripts are a copy of your performance at Punahou. So a transcript is a copy of some kind of information. So I actually didn't pick these images. I'm not super into like Mordor or whatever. I know this is like Lord of the Rings or whatever. Okay, so if we think of information in a textbook and transcribing that information, that's where we can maybe make a relation to this word, okay? So using this analogy in molecular terms, the DNA is the textbook and the mRNA is the notes. And then lastly, this information is translated. Presumably all of you are in some sort of foreign language and you do a lot of translating. which is literally one language to another. And in this analogy, this information is translated from a written to a spoken language. Okay, not exactly the same thing as like, you know, Hawaiian to French or something like that, but the analogy is an okay one. Okay, in our molecular terms, this translation is from the language of mRNA into the language of a protein. The thing that does the translating is the ribosome. The ribosome does that job. Okay. You don't need to worry about, you know, the central dogma or wrapping your brain around doing research there, but the central dogma is this. Okay, DNA to RNA to protein. The central dogma means it's a central idea, and it means that it's a very important, sort of unshakable notion that has a lot of research behind it. It was coined by actually one of the guys who elucidated or figured out the structure of DNA. And before we had a lot of information to back it up, he said the flow of information is always going to be in this direction. And he was right. So I'm not going to take us through this here, but you should click on this link to watch this video, which explains the overall process. Okay, probably at several times watching my video, you're going to say, gosh, she's repeating herself. She's making me reiterate this. Yes. Okay. One of the core ideas of neuroscience is this: repetition, repetition, repetition. Ha ha ha. Do you get the irony there? Okay, so repetition is the way in which we learn. We learn through repetition. So I repeat myself a lot. Okay, that's what helps you learn. Okay, so this central dogma, which I've repeated here, simply represents a flow of information. Okay, the analogy I like to use, I ran track for a really long time, and I really love the relays. So I don't know how many of you are familiar with that. One runner passes a baton on to another, who passes a baton on to another. So that's a good analogy. Okay. The DNA doesn't transform into an mRNA molecule, nor does the mRNA transform into a protein. Okay. In a relay, it's actually illegal for one runner to keep running in the place of another. Okay. So this is the central dogma, or this representation here is not showing you that these molecules change form into one another. Okay, it's just representing that very important information is passed along from one form into another. And this is essentially what we're looking at today. What are the processes that underlie this information passage? Okay, here's another repetition. Okay. So we have not yet learned about DNA replication. That's simply DNA making a copy of itself. You can see that right here. DNA making more DNA. It's super cool, super complex. We don't really need to know it right now. We'll learn about it when we're about to learn about cells making more cells, because that's when it happens. So we'll save that, okay? So kind of put this on hold here, and let's just focus on information being passed from DNA to RNA to protein, okay? Because the whole idea here is, what is DNA for? Okay, that's the name of your handout, right? Unraveling the Mysteries of DNA version 2.0. What is it for? What's DNA for? It's your genes, it's the information that makes you up, but that information really is just a code to build proteins. That's really all it is. It's a lot more than that, but that's sort of the business end of DNA. Okay, so here's a really basic diagram And it introduces to us the concept of gene, which I believe, I don't have it in front of me right now, but I believe we'll talk about next class. I would be willing to bet every single one of you has heard of a gene. You're going to refine your understanding of what a gene is, which is great. A gene is just a section of DNA that codes for a specific protein. Boom, there it is. You'll have to put it in your own words, of course. But I wanted to show you this because it's a really nice, simple image. Has a little bit of sort of orange in it. And it starts to get you thinking about the fact that just because we can think of genes as being sort of singular, it's not that simple. And I really like to get that idea across early, especially this part right here. Okay, some genes are expressed only at certain times or under specific conditions. When we talk about that, I'll actually give you a personal example of myself. Okay, so here's another Pear Deck question. So if I enable it for you guys, you can do it. We got a cool orange star and a red circle. Nothing against red, but the orange star is what you want to drag to the correct answer. Hopefully by now you know which is the right one. Okay, if you're not sure, ask me. I'm going to move along. Okay, and then RNA, we're going to start looking at all the different types of RNA. There's actually 3 main ones that we'll look at. You will go on later on to find out that perhaps that there's more than that. Okay, so RNA stands for ribonucleic acid. You guys have already learned about the similarities RNA shares with DNA and some of the differences it has from DNA. Okay, so I'm not going to go a ton into that. Probably the main one is it has uracil instead of thymine. Another couple things are it's single-stranded and it has a different sugar. Okay. Oh, I just named them there. I totally forgot that. Do I have something else here? Oh, okay. And then there's 3 types. Okay, so mRNA, tRNA, and rRNA. What you're seeing here is sort of like this is really sort of like a prototype one. Maybe it wants to be mRNA, but we'd have to have more information. Okay. So here's mRNA. Messenger RNA. Remember, everything is named what it's named for a reason. What is messenger RNA? Yep, it's a messenger. Okay, so it doesn't just store information in its sequence from DNA. Okay, it doesn't walk around or float around already intact. What you hopefully have learned from your activity is messenger RNA is constructed from a DNA template, right? So it's copied from the DNA sequence. What I'm going to start just using the terminology, it's constructed from a gene, right? A specific sequence of DNA. So it is then acting as a messenger, taking information from the DNA in the nucleus to where the DNA cannot go, to the ribosome. Okay, so if we look at this sort of shape of mRNA, what's important about it is the sequence of bases. Where did it get that sequence? From DNA. Okay, and this is sort of a simplified image of how it happened. So here's the double-stranded DNA, which is blue. It temporarily unwinds, revealing its sequence of bases. You can even tell which is the DNA. I wouldn't even have to tell you. How do you know that? How do you know that? Think about it. Think about it. Right, it has the T's, right? DNA has the T's. And then the nucleotides, the free nucleotides that you guys should remember from your activity, come in and one by one base pair. Okay. So then it forms this long strand of mRNA. If you're interested in what this 5' and 3' mean, those have to do with the carbons in the sugars in these strands, and I can tell you more about that if you're interested. Okay. The next RNA we're going to look at this random arrow. How fun. The next one we're going to look at is called transfer RNA. Yes, it transfers stuff. Maybe not the best name, but hey, I didn't name it. Okay, so transfer RNA, I would say, is the RNA that confuzzles and confounds and confuses students the most. So pay attention, okay? So transfer RNA is RNA, but it's all folded up. So it looks different, and it has a specific job, and its job involves one part of its anatomy, and then it carries something that is only temporarily part of it. Okay, so, oh, sorry, I should have advanced this a bit more. Okay, so it folds up by actually base pairing with itself, and this allows it to carry an amino acid. Okay, remember we talked about amino acids being the things that make up proteins. So tRNAs are the really important things that deliver amino acids in order for proteins to be built. Okay, but amino acids are just something that tRNA carry. They're not part of a tRNA permanently. Okay, the analogy I like to use is the tRNA is like a worker with a wheelbarrow, and the amino acid is the stuff that they fill up their wheelbarrow with. Okay, but you have to imagine the wheelbarrow is part of their body. Okay, so not the best analogy. The anticodon is part of their anatomy. We'll get to that in a minute. Okay, sorry, these little clicks don't work super well. We will see why this is significant soon. Okay, so here's a picture. And we got some arrows. Shoop, shoop, shoop. Or is that okay? Great. So these are some like a little more molecular pictures, and this is more of like a space fill model. Okay, so if you look really closely, you can see the bases, right? A's and C's and U's and stuff. Don't worry about ester bond, and they're really cool, okay, but it's some chem stuff that we can talk about if you want. So this is a long strand of RNA, but it's all folded, okay? And down here on the bottom is the important part, the anticodon. It's always 1, 2, 3 base pairs, okay? And this arrow is trying to point to it. I guess I didn't do a great job of that. It's probably because I used a blue arrow and not an orange one, okay? So after it folds, they really do kind of look like this shape, okay? And then you can see up here the amino acid just bonds to the 3' end, to one end, but it will also be dropped off, and then a new amino acid can be picked up. The job of this tRNA is to transfer an amino acid from wherever it picks it up to the ribosome. That's its job. That's it. A transfer RNA follows orders. That's it. Okay? All right. The last RNA we're going to talk about, there's another random arrow, is called ribosomal RNA. You guessed it. It helps make up ribosomes. It's that gold stuff. It coheres, it combines with with protein, and it makes up this really, really complicated molecule. Truly, a ribosome is one of the most complicated things in a cell, which we could talk about too if you'd like to. Okay, so the gold stuff in here is RNA, and the bluish purplish stuff is protein. So think about it just for a second, just for a second. Okay, to make proteins, which is what this thing does. Okay, this is a ribosome. You need protein. Isn't that cool? That's kind of cool. Okay, so anyway, so we're never going to talk really about rRNA. Like some students say, and then the rRNA makes protein. Okay, that's not accurate. The ribosome is the thing that makes proteins. Okay, the ribosome has rRNA in it. Okay, so you're not going to really talk about rRNA by itself. It helps to make up ribosomes. Talk to me about that if I just confused you. Okay. So let's look at the first step. Okay, now we're ready to look at step 1, transcription. And in transcription, we're taking the information that's in, so sorry, there goes my phone. It's my mom. Okay, we're taking the information that's in DNA and transcribing it into a messenger molecule. That's transcription. Okay, so where does it occur? In the nucleus. How does it occur? DNA unwinds. I'm going to add one little thing here, one step. Okay, this big sort of, honestly, it looks like a guitar pick to me. This is an enzyme. It's actually a protein that helps this process along. Do you see this right here? RNA polymerase. Okay, that's the name of it. You don't have to memorize it. You don't have to worry about it. Okay, but it's just so you start to understand, because lots of times students get confused. They're like, wait, the DNA just unwinds? It just happens? No, it doesn't. Okay, there's a lot of things that have to line up to make this happen. And this is actually only showing a very simplified form. There's probably 8 enzyme complexes that help this process along. It's pretty complicated. Okay, but when something ends with ACE, A-S-E, that tells you it's an enzyme, and almost all enzymes are proteins. That was actually in your handout that we're working on right now. Okay, so the DNA unwinds with the help of RNA polymerase, and these RNA nucleotides are built up into a strand of mRNA. And actually, that's what polymerase does. Poly means many. A polymer is a long molecule. So that's what this enzyme is helping do. It's actually a different enzyme that unwinds DNA, but we could talk about that if you want to. Okay? So here's another image. Repetition, repetition, repetition. So here's RNA polymerase again. Okay. The DNA is intact here. Nothing's really happening. Okay, we're just sort of getting it started. Now the DNA is unwound. Okay, notice it's just a part of DNA. It's just a specific sequence that codes for the protein we're going to make. That's a gene. And then this doesn't really show single nucleotides of RNA building this mRNA, which I don't love, but it does say it here. Okay. The reason I'm being so specific in all of my explanations is because I do expect your revised and amended hypotheses to be very specific. Okay. Translation. Translation, remember, we are translating the information that we have in mRNA into the language, first, of amino acids, which will then be put together into a protein. There's a little trick to this. So again, you gotta shake yourself awake and pay attention. This is where the anticodon that I showed you on the tRNA matters. And then we have to also learn about codons in mRNA and see how those all line up. Okay? So we already kind of looked at how the sequence from the DNA, the gene, is now contained as a sequence of bases in the mRNA. Okay, so we basically got a sequence of bases. What scientists discovered, and this was like such a huge discovery, the way they did it was so cool. For a long time, they knew that ribosomes translated RNA, but they didn't know how. So they finally figured out that ribosomes actually read, okay, you can't see my fingers, but I'm doing the little air quote things. They actually read mRNA in groups of 3. So the mRNA is literally pulled through the ribosome, and there's a specific sequence that always begins a protein, and that is AUG. That's always going to be the first codon as a signal. Okay, we're starting a protein, and it's methionine. That codes for the amino acid methionine. One 3-letter sequence is called a codon. Okay, that was just like a word made up by the dude who discovered this. It was a dude because, okay, we could get into a long conversation about like the history of science and gender equity and stuff, but we won't. Okay, so they're called codons, and every subsequent 3-letter sequence is also called a codon. Okay, so the ribosome has like a reading frame, right? It reads 3 at a time, the next 3, the next 3, and each one is coding for a specific amino acid. Okay, great. So where do the tRNA come in? Well, that's the beauty of base pairing, okay? All of this rests on base pairing. Okay, back up for a second and think about it, right? We got mRNA because it base paired with DNA, right? Let's go back for a second. The way that we built our RNA was by base pairing. Now, the way that we're going to build our protein is by allowing tRNA's anticodon to base pair and double check to see if it can correctly drop the amino acid it's carrying. Okay, the reason for this is what I tried to emphasize before. The tRNA is only following directions. It never talked, here again I'm doing air quotes, to DNA. Only the mRNA communicated with DNA. So here's what it looks like. Here we have our mRNA, which actually contains the transcribed message. Okay, mRNA knows the deal. It talked to DNA. mRNA is saying to the ribosome, which I know is not pictured here, this is the message. Okay, translate this. So the ribosome is calling tRNAs. Okay, and each tRNA has its anticodon, which corresponds to whatever amino acid it's carrying. And the tRNA is just saying, okay, I'm carrying this one. Is it the right one? So the way it checks is by base pairing. And if it can correctly base pair, okay, you check yourself. Are these correctly base pairing? GC, AU, GC. Remember, RNA does not have T. Okay, so yes? Yes? Do you agree with me? I think that yes. If these correctly base pair, and the ultimate decider is the ribosome. Okay, the ribosome decides if this is correct. Then the tRNA will drop its amino acid, and then, boop, it leaves without it, the amino acid, because it dropped it off. And then it goes and does its job again. It goes and gets another amino acid, and it could go to a whole different ribosome and help build a different protein or come back to the same one. Okay, because you have millions of ribosomes building billions of proteins at any given time. Okay, so hopefully that makes sense. This is kind of a highly simplified image of translation. Okay, so what's happening here? This blue thing that looks like a bread slice of bread, is the ribosome. It actually exists in two pieces. So the two pieces kind of clamp on to the mRNA, and it reads the mRNA, remember, in codons, in groups of 3. And what you can see is that these tRNAs are coming in, right, matching up their anticodons. Okay, so what's happening here is this one just released, I think that's glutamine. Okay, these two are tRNA that have already released their amino acids, glycine and serine probably. So these ones are going to go pick up more. That's why they're called empty, quote unquote. These ones are coming in, right, ready to base pair. You can kind of anticipate where they would base pair. This is not accidental. Okay, there's all kinds of signaling methods that the ribosome has to sort of call these tRNAs to be sort of lining up, because protein synthesis is, important business. All right. There's also a link here for an animation that you should watch. Repetition, repetition, repetition. Do yourself a favor. This is complicated stuff. Okay. If any part of this doesn't make sense to you, please let me know. All right. So from our activity, this is sort of, I like this image because sort of like the first step that we did. Okay, stuff wasn't necessarily really labeled. It was just shapes. I like this a little better because the uracil is its own color. We didn't have that in the activity, plus it's orange. And this also shows RNA polymerase, which I'm sort of trying to get us used to, right? As the DNA is not just mysteriously unwinding, there's something that helps this process along. And here's the mRNA forming. Okay, so this is transcription. This is the information going from DNA to mRNA. All right, that's the first step. Here's the second step. So kind of now, once we have the messenger RNA molecule, it leaves the nucleus, comes to the ribosome. The ribosome is reading it in groups of 3. Here are the tRNA molecules coming in with their amino acids. Once their anticodons are base pairing with the codons in the mRNA, then their amino acids can be dropped off and they can leave and go get another amino acid. It is the ribosome that organizes and mediates and is in charge of all those processes, which is why the ribosome is so complicated, super complicated as a molecule. It's also the ribosome that takes these individual amino acids and actually bonds them together and makes this polypeptide chain. It's actually not technically a protein yet, which is why you see this step. Okay, but close enough. We call it a protein, okay, because we're not going to really talk about all the steps it goes through to become a protein, unless you want to. Okay, so you guys actually had a stop, a tRNA carrying a stop signal. So we'll talk about that, right? Because it doesn't just go on indefinitely. The stop signal is actually, of course, coded into the DNA. It's the end of the gene. And what actually happens is let me back up a second. That stop code that's encoded into the DNA, of course, is transcribed into the mRNA. And that codon will be one of these 3, UAG, UAA, or UGA. And when one of those is transcribed, then they will be translated, of course, and the ribosome will read that. And then a special tRNA will come in and will signal the end of the production of that protein. And then what actually happens is the ribosome itself lets go of the mRNA. And depending on what some of the other signals are, sometimes that mRNA will actually go and be translated again. Lots of different fates can happen, but the mRNA is always either translated again or recycled. It's chopped up and the nucleotides are used again. Okay, so let's look a little bit more at overall process. Okay, so starting from we're assuming we knew that what the deal was with transcription, right? We got that down. So here's translation, right? With the ribosome already clamped onto the mRNA. We know, or we're starting to understand that methionine is always the first amino acid with this AUG here. Okay, and then here comes the next one. Then these amino acids are being put together by the ribosome, and then we have a growing boop, boop, boop chain of amino acids to make this longer protein. Okay, and then finally there's a stop. AUG is a stop codon, and that's gonna pull the ribosome off of the mRNA, and then we've got this long chain of amino acids, which is accurately called a polypeptide. There's that prefix again, poly, meaning many, and peptide refers to this peptide bond here. It's the kind of bond that is made between amino acids. Okay, so what I would like you guys to do on this pair deck is just practice. Okay, if these are the codons, what are the anticodons? It's just base pairing, so you should be able to whip this out, right? Base pair, base pair, base pair, base pair, base pair. Don't race, but you should be able to do this. You will do this again. This is in your future, so don't skip it. Okay, I'm gonna keep you honest. I'm not gonna give you much time because you can pause me and then I'll be back. Okay, so in order to determine what amino acids are being sort of called for by the mRNA, there is a codon chart. Okay, 500 million gazillion quintillion times I will tell you. You use this chart with mRNA. Why? Well, mRNA is the molecule that transcribed the message from DNA, not tRNA. Okay, still choke times, like lots of times. Someone is going to be doing an activity in class and will accidentally translate the anticodons. Okay, don't do it. So here's a link here guiding you how to read a codon chart. You can watch that. But I think I also have my next slide. Yep. Okay, so let's say in our first codon is always AUG. Okay, again, we're talking about mRNA, not tRNA, okay, not the anticodons. So the simple way that you use this chart is you just go first, second, third. Okay, so my first base is A, so I know it's got to be in this row. Okay, my second one is U, so I know it's got to be in this column. And then my last one is G, so I just triangulate. G is right here, methionine. Okay, pretty simple. And there's also let me know if that's hard for you. Okay. Oh, I have a pair deck for you. Sorry. So for the, oh yeah. Okay. So all I wanted you to do for this one is I'm giving you actually a DNA strand down here. Okay. So the first thing I want you to do, two things, right? Using this DNA strand right down here, first thing I want you to do is transcribe it, okay, into mRNA, and then figure out the amino acids. Okay, keep in mind, you don't need to do the tRNA anticodons. You don't actually have to do that. You can, but all you really need is your mRNA, and then figure out the amino acids. Okay, if you get to this point and you're like, what, then stop it and let me know if you need help. Okay, so here's the answers. Okay. So there's the transcribed mRNA sequence, right? And the way that you derive these is, again, by using this chart. And yes, I'd like to emphasize tRNA is super important because it delivers these amino acids, but you don't have to actually show the anticodons, right? Not that they're not important, but they're sort of inherent in the delivery of these amino acids. Okay. What I thought I was going to say a couple slides back was this is another version of that chart. It's the circular version. To my knowledge, there's no like official name. But for, let's say we were doing methionene's codon, we would go start in the middle, A, U, G. So you always start in the middle and work your way out, right? First, second, third, and then you'd end up at methionine. Okay? I think this is the last slide. So what am I going to do? I'm going to ask you to repetition. That doesn't make sense. I'm going to ask you to repeat. Okay, so here's some videos, lots of videos, and there's actually some practice on Canvas transcribing and translating genes. Okay, let me know if you have any questions, and have fun.